Hello everyone. Let's study dihybrid cross. Now, what is the dihybrid cross? It's basically a cross between two plants which are pure and contrasting with respect to two pairs of traits. Or we can say we simply consider two different characteristics while making this cross. We can either say two characters or two pairs of contrasting traits. Let's understand the dihybrid cross with an example. Now, this is the same cross as given in your textbook. Let's take the same cross. Here, we consider two characteristics from the garden pea plant. That is, first we are considering the seed shape. In case of garden pea plant, seed shape would be round or wrinkled. Next character is seed color. Seed color. It could be yellow or green. So here you can see we have taken the parent. One is round yellow seeded and the second one is wrinkled green. So both the parents are contrasting. Also look at the genotype. First one is capital R, capital R, capital Y, capital Y. So for both the genes, it's homozygous condition. Identical pair of alleles are present. Capital, capital, small, small, right? So this is homozygous dominant for both the traits. Now see the other parent, small r, small r, small y, small y. So this has wrinkled and green seed. What we can see here, this is also homozygous. It is a pure line because again same copies are repeated. R, small r, small r, small y, right? So as we have seen in the monohybrid cross also, we basically use parents which are contrasting and pure lines. So here also we have taken two parents. Both are contrasting because they are having opposite characteristics. See, this is round, this is wrinkled. Next, yellow, green. So, contrasting and both are pure, both are homozygous. Now, the first step here will be to form gametes. So, as here, both the characteristics, both the genes which you can see are pure. So, only one type of gamete will form. How? Let's understand. See, here we have the parent capital R, capital R, capital Y, right? According to law of segregation which we have studied in the monohybrid cross, right? This RR will separate and this Y will also separate. Okay, during gamete formation, this pair of alleles separate. So RR will separate and Y will also separate. Now how they will separate? This first R can move with the first Y or with the second Y. There is equal chance. Next, the second R can move with the first Y or with the second Y. So, in this way, you form any combination, you will get only one type of gamete which is capital R, capital Y. That is why here you can see we have mentioned only one type of gamete. Similarly, for the second plant with wrinkled green seeds, here also we have only one type of character. See, here R will separate, Y will also separate. Any R from here can move with any Y. You take any combination, you will get small r, small y combination in the gamete. So here also only one type of gamete is produced. Now these gametes will fuse and you will get the first filial generation, the F1 generation, which showed characters like this, round and yellow. Okay, the seeds in this generation showed round shape and yellow color. So as we are seeing here, there is round and yellow characteristics expressed in all the F1 plants. That is why we say round and yellow. These are dominant traits. Okay. And the opposite, wrinkled and green, are recessive as they are not expressed here in the hybrid. See, in this cross so far, we took the contrasting and pure lines, made a cross between them. We got all these plants in the F1 generation. All the plants which you get in F1 generation, okay, let's say you get 2000 plants. All the 2000 plants have round and yellow seeds. Okay. So, now let's look at the genotype. What's the genotype here? Capital R small r, capital Y, small y, because these two gametes were involved in fusion. So genotypically what we see here is both R and Y are heterozygous, right? Heterozygous combinations. But only one of the parental characters is expressed, that is round and yellow, right? Now, what is the next step? We have got the F1 generation. The next step is selfing of F1 generation plant to obtain the F2 generation, that is second filial generation. So, let's see the next step. Okay. So, F1 generation, we already have capital R, capital Y, small y. Now, we will do the self-pollination. Okay. As the pea plants, these have bisexual flowers. You can easily do self-pollination. So both male and female gametes will be provided by the same plant. Okay. Now, what are the different types of gametes which will be produced in this F1 plant? Okay. So let's write the selfing step. Capital R, capital Y, small y, cross with capital R, capital Y, small y. Right? Same plant is being used, like both the gametes, male and female, are providing the gametes, right? Same plant is providing both the gametes, male as well as female. So, how many different types of gametes will be produced here? Now, to solve this, let's try to understand. We have capital R, capital Y, small y. Okay? Law of segregation. So, R will separate, Y will also separate. Okay? From each other. Law of segregation. Here, this capital R can move with capital Y or it can move with small y. Equal chance, 50-50. Next, small r can move with capital Y or with this small y. So, now, how many different combinations can you write? Here, we can write 4, right? So, gametes produced in the F1 plant. Let's write. First, we have capital R, capital Y. Next, we have capital R, small y. Next, we have small r, capital Y. Okay. And last, we have small r, small y. So, these are the 4 possible gametes. Let's say these are male gametes. Then, the same plant is providing female gametes. So, the female gametes will also be of same 4 types. So, let's write it. Female gametes, same. Capital R, capital Y. Capital R, small r, capital Y. And small r, small y, right? So, 4 types of male gametes and 4 types of female gametes. So, 4 into 4, we will get 16 different offsprings in the F2 generation. Okay. Now, to represent all the possible types of gametes and all these 16 offsprings, okay, we simply make a table which is called as Punnett square. It's very easy way to represent all the possible gametes and all the possible offspring. Okay, so let's draw the Punnett square and find out what will be the offspring in next generation, that is F2 generation, second filial generation. So here let's write all the gametes and the possible offspring. Okay, so let's say first let's write the male gamete and here vertically we will write the female gamete. Okay, first we have capital R, capital Y. Next we have capital R, small r, capital Y. And next we have small r, small y. Okay, let's see, these are the male gametes. Now female, we have the same 4 types. Capital R, capital Y, capital R, small r, capital Y, and small r, small. These are the gametes, male and female gametes. Okay. Now, here you can write these gametes in any sequence. Okay. This is not some process that you have to write it in a fixed sequence, but it is easier to basically understand how the offsprings are and to remember the different possible offsprings if you write the gametes in this sequence. Okay. So, make it a habit that you write gametes in this sequence. Okay, doesn't matter if you don't write, you will get the same ratios in the offspring. But if you write the gametes in this sequence, then the offspring which you will get, it will show a specific pattern. Okay, and it becomes a lot easier to remember how many are homozygous, how many are heterozygous offspring. Okay. So when we will write all the genotypes, you will understand this. So now we have written all the gametes. Next step is to find out all the possible offspring. So how to find out the offspring? Very easy. If let's say we take this first female gamete, okay, we have this first female gamete. It uses with this first male gamete, we'll write it here. Okay, let's say this first female gamete uses with the second male gamete, we'll write it here. Next, the first female gamete using with the third male gamete here, we'll write it here. So in this way, simply do the match the pair and write all the possible offspring. So one by one, let's write all the offspring. So capital R, capital R, capital Y, right? This is the first offspring. Next, capital R, capital R, capital Y, small y. Okay, this and this. Okay, just merge the genotypes together. Next, we have capital R, small r, capital Y, capital Y. Okay, next we have capital R, small r, capital Y, small y. So in this way, you have to fill the entire box. Let's complete this box quickly. We have capital R, capital R, capital Y, small y. Next, capital R, small y. Next, we have capital R, capital Y, small y. And here we have capital R, small y, small y. Okay. Next, we have capital R, capital Y. Next, we have capital R, capital Y, small y. Next box, we have small r, capital Y, capital Y. Okay, in this way, you have to just match the pair. Okay, next we have small r, capital Y, small y. Right? Next, last row, capital R, capital Y, small y. Next we have capital R, capital Y, small y, right? Undo this, and we have this. So we will have small y. Next we have small r, capital Y, small y, and last small r, small y, small y. So in this way, we have filled the entire unit square. Okay. We have written all the genotypes. Now we have to find out all the phenotypes of offspring. Okay. Now simple. If we have even a single copy of capital R, the shape of seed will be round because we know round is dominant character. And even if you have a single copy of the allele, it will be expressed. Right. Next, if you have capital Y, then yellow color. Okay. If you want wrinkled or green character, then we should have homozygous recessive combination. Okay, so one by one, now let's fill all the possible phenotypes here. First, we have capital R, capital Y, capital Y. So in this case, as capital R is present and capital Y is also present, the shape will be round and color will be yellow. So round yellow. Next, again capital R is present, capital Y is also present. So round yellow. Next, round yellow, right? Next also round yellow. Next, capital R is present, capital Y also, so round yellow. Now see the next one. Capital R. So round is confirmed. What is next? Small y. Homozygous recessive gene combination. So here the color will be green. So round shape and green color. Next we have round yellow. Next we have again round green. Next we have round yellow. Okay, I hope this is clear. Simply look for capital letters first. Capital R is present, so round. Capital Y, even if single copy, then yellow. Okay. Next, now your small r means wrinkled shape. And capital Y is present, so color will be yellow. So we have wrinkled yellow. Next, same here, wrinkled yellow. What is next? Round yellow. Next, round but green. Right? Round but green. Next, we have, it will be wrinkled because of small r, and color will be yellow. So wrinkled, yellow. And next, last, small y. So it will be wrinkled green. Recessive characteristic for both the characters. Okay, recessive for both. We have wrinkled and green. So here we have filled the entire Punnett square, all the genotypes of 16 offsprings and also the phenotype. What is the next step? Yes, basically this is the F2 generation, right? All these 16, these are F2 generation offspring. So now we have to find the ratio. Okay, first let's find the phenotypic ratio. Phenotypic ratio. For phenotypic ratio, obviously we will consider only the physical appearance of the traits. Okay, we will not look at the genes. Based on phenotypes only, we will write the ratio. Now, in these 16 offspring, we have different traits. Okay, different combinations of traits. So first, let's write all the possible combinations. Okay, first we have round shape and yellow color. So round yellow. Next, let's say we have round shape but green color. So like this. Okay. Next, we have, let's say, wrinkled shape but yellow color. And last, we have wrinkled shape and green color. So this is the combination, right? So we have four different phenotypes in F2 generation. Round yellow, round green, wrinkled yellow and wrinkled green. Let's count how many round yellows are present. Okay, so we have 1, 2, 3, 4, 5, 6, 7, 8 and 9. Right? So we get 9 round yellows. Next, we have round green here. 1, 2 and 3. So total 3. Next, we have wrinkled yellow. You can see 1, 2, 3. Right? These 3. So we have 3. And last, wrinkled green, we have only one, this last one. So this becomes our phenotypic ratio. 9 is to 3 is to 3 is to 1. If you make a dihybrid cross, then in F2 generation offspring, you get phenotypic ratio 9 is to 3 is to 3 is to 1. 9 plants are such that it shows both dominant characters. 3 plants show, let's say, first dominant, second recessive. Next 3 plants, first recessive, second dominant. And last 1 plant, we have both recessive characters like wrinkled green. So this becomes the phenotypic ratio. Now next is genotypic ratio. Okay, like we also calculated a genotypic ratio for monohybrid cross. Now let's do it for dihybrid cross. Let's find out the genotypic ratio. Genotypic ratio. So now one by one we will also list all the genotypes and count how many plants are present with respect to the given genotype. We have to see how many different genotypes are present and number for each. Okay. So let's start with the first one. Okay, we will go in sequence. The first one we have capital R, capital R, capital Y, capital Y. If you look at the entire Punnett square, how many plants are present with this genotype? Look at all the genotypes. Then we see there is only one, the first one. Correct? So here we have one. Okay? So this is done. I will just add a tick so that we understand it's done. Okay? This is done. Now, next one, capital R, capital R, capital Y, small y. Let's count how many plants with this genotype. So obviously this is one. Do we have more? Yes, here. Correct? So we have 2. Can you see more? No, we have only 2. So 2. Now what is the next one? Capital R, small r, capital Y, capital Y. Let's count how many with this genotype. Capital R, small r, capital Y, capital Y. This and here. Correct? So we have 2. Next, see this one? Capital R, small r, capital Y, small y. Okay, how many? Here you can see 1, it is 2, 3, and 4. These, like, right? Can you see it? Diagonal, like this. So we have 4. So there are 4 plants out of total 16 which are heterozygous, hybrid for both the genes. Okay, let's see the next one. This one, this is done. So this one, okay, capital R, small y. Okay, this one, is there anywhere else? No, we have only one. Okay, let's see the next one. This is done. So this will be the next one. Capital R, small y, small y. Let's count how many with this genotype. This is one. So we have more. Yes, here, right? So we have two. Next, let's go into because this is done. This is done. So now this one. Small r, capital Y. Okay, count how many with this? Only one. So one. Okay, next. So this is done. This is next one. Now this one. Small r, capital Y, small y. Count 1 and 2. So we have 2 and now last this one, just one, right? Small r, small y, small y. One. So this is our genotypic ratio here. 1 is to 2 is to 2 is to 4 is to 1 is to 2 is to 1 is to 2 is to 1. Total 9 different genotypes. Count 1, 2, 3, 4, 5, 6, 7, 8, 9. Total 9 different genotypes are present in the 16 offspring, and these 9 genotypes are present in this ratio. Okay. Now, many numerical based questions are asked, like some dihybrid cross is done. Then in F2 generation, how many plants, okay, are present which are hybrid for both? Okay. Now that time you can't waste your time in your exams by drawing the entire table and figuring out how many are hybrid for both. So that time this genotypic ratio and remembering this will help. Okay. So like here you can see hybrid for both. This one. Okay. Capital R, capital Y, small y. Hybrid for both. There are 4. Right? So this is what you need to analyze while making the cross. Okay. Now the question. Okay. Try to answer this question. Out of these 16 offspring, how many offspring are homozygous for both the genes? Okay. Homozygous for both the characters. Okay. Try to find out first. Homozygous for both the characters. Okay. Now see here. This is obviously one. Correct? See this also capital R, small y. This is also homozygous. Okay? Okay. First is homozygous dominant, second is homozygous recessive. But it is homozygous. The question was homozygous for both genes. So we have to count this also. Next is this and this. This is obvious one. Last one. Right? So again this diagonal like this. 4. Out of 16 there are 4 which are homozygous for both the characters. Right? So that you can see each is like 1, 1, 1. See here. Homozygous for both 1. Homozygous for both 1. Homozygous for both 1. And this last 1, 1, 1, 1. So this is how you can easily remember. Now what is remaining? We are done with all the ones. We are done with this 4. Now what is remaining? Only 2, 2, 2, 2. So easy. Homozygous, heterozygous. Heterozygous, homozygous. Right? See this. Heterozygous, homozygous. Homozygous, heterozygous. So simple. If you have like any one gene homozygous and other is heterozygous, like first heterozygous, second homozygous, whatever, dominant, recessive, then it will be 2, 2, 2, 2. Right? So this is how you can easily solve any numerical based question. If you already know that out of 16, you will have 4 plants which are hybrid for both. Right? We will have only one plant which is homozygous dominant for both. Right? So This is the entire Punnett square representing all the possible gametes of F1, male and female, and next all the possible offsprings in the F2 generation. The phenotypic ratio is this 9 is to 3 is to 3 is to 1. And then the genotypic ratio is this one. Okay, remember this. 1 is to 2 is to 2 is to 4 is to 1 is to 2 is to 1 is to 2 is to 1. Right? Very easy. In your textbook, basically, this is how all the possible gametes, male and female, and the offsprings are given. Okay, now as this is a little tilted, so our diagonal basically will shift now. In this case, you can see these four like this here. These four are heterozygous. Correct? Capital R, capital Y, small y. These four are heterozygous. And if you look at these four, these are homozygous for both. Right? These are homozygous for both. Okay, so this is how you can easily understand the dihybrid cross. Now, when we looked at the dihybrid cross, okay, we always saw whatever the genotype is. Okay, now whenever we wrote something like this, let's say first, like pure line, pure parent, we always applied the law of segregation. Okay, it is universal. So this R will separate and Y will also separate. Okay, and a gamete will receive only one copy of each gene. So R one copy, Y one copy. So in this case, whatever combination you make, you get only one type of gamete, which is capital R, capital Y. Okay. Next, we saw if we let's say take a hybrid. Erase this. Okay. Next, let's say when we took hybrid, like this. Always R separate, Y separate, right? And you form the possible combinations like this. So you get four different types of gametes as we saw the gametes of F1. Now what you can see here is it's not something like now this plant phenotypically was with round and yellow seeds, right? Round and yellow seeds, but it definitely had small r and small y, okay, in it, but it is not expressed, and that is why we called it recessive, right? When gametes were produced, basically segregation of R and Y, whatever, dominant or recessive, it was always independent. Okay, it was not like capital R, so I have to go with capital Y. Was it like this? No. Capital R can move with capital Y as well as small y equally. Same way, small r can move with capital Y or small y equally because the chromosomes and the genes present on these different chromosomes assort independently. And as in dihybrid cross, we consider two characters. So we have two different genes like R and Y. We can easily explain the independent assortment, like law of independent assortment based on the dihybrid cross. So this is very important. See, the first two laws, law of dominance and law of segregation, could be easily explained with a simple monohybrid cross. Okay, but in monohybrid cross, we consider only one character. So we can't explain the third law with monohybrid cross. Okay, in independent assortment, basically we explain how the genes controlling two different characters assort independently. Like in the dihybrid cross here, we took seed shape and seed color, right? We have a different gene for seed shape, we have a different gene for seed color, and they always separated independently. So, based on the dihybrid cross result, we get the Mendel's law of independent assortment. Okay, and according to this law, what can we say? See, the law states that when two pairs of traits are combined in a hybrid, like we saw in capital R, capital Y, small y, right? The F1 generation plant. When two pairs of traits are combined in a hybrid, segregation of one pair of characters is independent of other pair of characters. So segregation of this capital R and capital Y small y is independent of each other. Okay, see, when R is separating from each other or Y are getting separating from each other, this is law of segregation. But separation of this R and Y is independent. This is the third law. And as here we have to take like minimum two different characters. So law of independent assortment can be explained only on the basis of dihybrid cross. Okay. So in this video lecture, we have studied how to make a dihybrid cross, the ratios, phenotypic and genotypic ratio of F2 generation plant in dihybrid cross. And last, based on the dihybrid cross, we can explain the law of independent assortment. We have studied the law of independent assortment. Okay. I hope this video was helpful for you. In the next class, we will study linkage. Thank you.